We're going to talk about values and means in the population under a single locus model. So we are going to start with the generic values. Then we're going to use those generic values to estimate the population mean. And then we're going to talk about the average allele effects in the population and the average allele substitution effect in the population. So first, to talk about the generic values, we have to remember the genotypes and this Punnett square is just I think that it's gonna help you to remember that again, if we cross two heterozygous lines, remember that each heterozygous lines can transmit to the progeny two gametes. So in this case, uh, each of the heterozygous lines are going to transmit or the A1 or the A2 gamete to the progeny. So if we cross those two lines, we're gonna have the homozygous lines in the proportion in the frequency of p squared that are gonna be the frequency of the homozygous, and the same for the homozygous for the A2. So remember that we learn how to estimate the allele frequencies of P and Q, and the heterozygous lines are a 50% of the Punnett square here. So as you remember, using the formula of Harding-Wember, we have that basically the expected genotype in the population, it's going to be p squared, 2pq, and q squared. So that is why I mentioned before that we're going to talk a lot about p and q, and it's good that you remember those letters and the concept that they are the allele frequency in the population. So now I want to introduce you two new concepts that are the A and the D. And basically the A represent the additive effect of the genes. And here I'm just gonna talk about a single gene or locus. And D represent the deviation from the midpoint of the uh, homozygous line. So just in these examples, you can see that we have the phenotypes of both homozygous are homozygous A2, it's one flower, have one flower. So here we have the trait that we're using here are the number of flowers, and the homozygous for A1 have three flowers. So the midpoint between both homozygous it's 2, and here on using this model, so that 2 represents 0. So 0 it's the midpoint between both homozygous lines. And when the heterozygous lines it's different, it's not in the middle of the both homozygous, so we can say that there is a dominant or that deviation is caused because some degree of dominance. And in this example here, you can see that we have a partial dominance that is caused, that is due to the A1 allele. So basically remember that we have now the genotypes, and now we have the genetic values that are represented with A and D. And with those values, we can estimate the population mean. So we are going to use the same formula that we used in the previous video when we use the frequencies of the genotypes, and now we have the values or the genetic values, the genotypic values here. So as we have the formulas, the frequencies that are again p squared, 2pq, and q squared that represent the genotype frequencies in the population, we can estimate the contribution to the mean of each single genotype. So we have the contribution here that is p squared times a and that is the contribution of the homozygous A1A1, and we can do the same for the other genotypes. And at the end, when we add all the values or the contributions to the mean of the three different genotypes, we can get the population mean. And this population mean it's equal to the additive effect that we can estimate in the population times the difference between the allele frequencies in the population. So that is why allele frequencies are important to understand the effect of the genotypes in the population. And we have to add the heterozygous lines and the dominant deviation of that is caused because some degree of dominance in the genotypes. So as you can see here, if the dominant deviation is 0, so that means that if the heterozygous lines are right in the middle between the homozygous, so this part of the formula here it's 0, so we can see that the expected population mean it's equal to the additive effect times the difference between P and Q, that are the allele frequencies. But if there is some degree of dominance or deviation because dominance, the expected mean of the population is equal to the additive effect times the difference between the allele frequencies plus the heterozygous lines and the dominant deviation. So I'm going to show you an example just to give you a better understanding of this formula. And just imagine that we have two genotypes. So we're going to use the same example with flowers. So we have one genotype that have one flower that it's the A1, A2, A2, and this is a complete additive gene or a gene with a complete additive effect in the population, and we cross that plant with another one that have three flowers. So the plant that has three flowers, it's homozygous for A1, A1. So if we cross those lines, we're going to have an F1 progeny that all of the individuals of the 100% of those individuals are going to have two flowers. And if we advance that F1 to the F2, we're going to have a segregation of this trait, and 25% of the individuals are going to present one flower, 50% two flowers, and another 20% three flowers. So now uh, we can use the information that we have in the table basically to try to give you the understanding in how we can use the formulas. So first we need to estimate the allele frequency of P, and as we know what is the frequency of the homozygous here that are 0.5 actually for both of them, so we just have to root square that frequency and that is equal to 0.5, and then we have to subtract that P to 1 to estimate the value of Q. So now we estimated that the value of P and Q, that are the allele frequencies, are both of them equal to 0.5. And now we have to estimate the value of A and D in the population. So remember that I told you that under an additive effect, the heterozygous lines are assumed to be in the middle of both homozygous lines. So here you can see that the difference between the homozygous A2A2, between homozygous A2 and homozygous A1, it's two flowers because A2 have a one flower, A1 have a homozygous A1 have three flowers. So the difference between both those two homozygous lines are two flowers. So the midpoint between those homozygous is 2, but here we are going to represent that midpoint as 0, and then the difference between that midpoint and each homozygous is just one flower because again the difference between both homozygous. It's 2, so the half of that difference, it's just one flower. So with that information, we can estimate that the value of A it's equal to 1 flower. And with that information, we just can put the numbers in the table. So here we just put the allele frequencies, and here the value of 1 because that represents one flower here, and here we have a negative 1 because basically uh, we're gonna reduce it's the midpoint, so it's gonna reduce in the same units the phenotype. And at the same that we use the frequency to estimate the mean of the population formula, previously we multiply the frequencies in the population times the value of the population, and that is gonna be the contribution of each genotype to the population mean. And then we can add the contributions of all the three genotypes, and this is the population mean. So we have that one here is the additive effect, that is the difference between the midpoint and each one of the heterozygous lines and each of the homozygous lines, that is one flower. And then we have the little frequencies of the population times the heterozygous lines and the dominant deviation. So hopefully you can see here that if there is not a dominant deviation in the allele that you are trying to estimate the effect, the second part of the formula is going to be equal to 0. And here, as we have the same allele frequency that is 0.5 and for P and 0.5 for Q, this part of the formula is also equal to 0. So that means that the mean of the population or the population mean it's equal to 0. And maybe in this moment you think that I am wrong because all the lines at least have one flower, so the mean of the population cannot be equal to 0. But the point with this formula is that as we're assuming an additive model, that population mean is measured from the mid homozygous point, which is 2 flowers in this example. So basically this is going to be the deviation that we're going to have in the population mean from the midpoint between both homozygous. So as the midpoint is 2 and the mean of the population here is 0, so the mean of the population is going to be equal to 2. And we can use the same formula but now with the phenotypes, and as you can see here we can also estimate that the mean of the population is 2 as well. So both formulas give you the same outcome, that it's that the population mean is 2. And what happens if now we have a different allele frequencies in the population? So I want to point out that that is so important because with different allele frequencies, we're going to estimate a different population means. So I did the same that in the previous slide. I just changed the allele frequencies in the population, and now again this is again a gene with an additive effect. So that means that the second part of the formula as the deviation, because dominant is 0, so the second part of the formula is 0. So we just have to take into account for the additive effect and the difference between P and Q. And here you can see that the mean of the population is equal to 0.8. And again, that is a deviation from the midpoint of the homozygous genotypes. So the midpoint is 2, and now as the population mean is equal to 0.8, so we have to add those values, and now we have that the population mean in a population mean with these allele frequencies is equal to 2.8 flowers in the population. And that makes sense because if you see here the proportion, the frequency of genotypes that are homozygous for A1 and have 3 flowers in this population or in a population with these allele frequencies, it's equal to 0.81. So that means that 81% of the individuals in this population have 3 flowers. 18% of the population have 2 flowers, and just 1% of the population have 1 flower. So that is why the mean of that population uh, it's higher than in the previous example. And as I mentioned, this is a single locus model that the expected mean of the population is equal to the additive effect times the difference between P and Q plus the heterozygous lines and the dominant deviation. And if you are interested to basically estimate the effect of several loci at the same time, you have to estimate or you have to add the uh, effects of every single loci, a locus in the population, and then you just have to add those values to estimate the mean of the population. And now that we understand how we can estimate the population mean with the genotype and with the genetic values, we can talk about the average allele effect. So for the average allele effect, basically, or what is the average allele effect? So imagine that we have a population, and from that population, we have some individuals that have the A2 allele. So this population have A1 and A2 alleles. So we have one subpopulation that have the A2 allele, and the another allele come randomly from the population. And we have the same case but with another allele. So we have other individuals that have the A1 allele and another allele from the come randomly from the population. So the average allele effect is the difference between the mean of the original population and the mean of the population when we have the A2 or the A1 allele when the another allele come randomly from the population. So that difference of the means are defined as the average allele effect. So we have alpha 1 and alpha 2 and alpha 1 to represent that average allele effect. And again, it's important remember that the genotypes transmit the gametes to the progeny. So produce basically they transmit the alleles are not the genotype. So that is important, and that is why we mentioned meiosis in the first presentation. So here remember that if we have 2 alleles in the population, so the parent just can transmit A1 or A2 to the next generation. And then we have the genotypic values here, that is A and B. So to estimate the mean value of the genotypes that are gonna we can get with the gamete A1, we have to take into account the frequency of A1 times the additive effect that have the genetic value of the homozygous of A1. And also we have to take into account the frequency of Q in the heterozygous lines, because that have like an effect the not just the frequency of A1 but also the frequency of A2 in the heterozygous times the dominance deviation that we have in that gene. And we have to do the same for the A2 allele. So basically A2 it's in the as A2 is in the heterozygous lines, so we have to take into account the frequency of P, that it's another allele that are that is in the heterozygous lines, and we have to take into account the frequency of Q, that it's the frequency of the A2 that it's in the homozygous A2. And so we have to, that it's the mean value of the genotypes when we have A1 or A2 here in the table. And we have to subtract to estimate the average allele of effect of A1 or A2. We have to subtract the population mean to that mean values. And with that, after subtract that, we can estimate the average effect of the allele or the gene. But I don't want to make your life so complicated, so that is why we're going to mention, or we're going to use more the average effect of the gene substitution. And that gene substitution effect is the difference between alpha 1, that is this one, and the alpha 2, that is the effect of the another allele, and that is equal to the additive effect plus the dominance deviation plus the difference between Q and P. And that is why, again, we're going to talk about that substitution, allele substitution effect, and with an example, because I think that is easier to understand with an example. So as I mentioned, alpha, that is the allele substitution effect, it's equal to the additive effect plus the dominant deviation, and the difference times the difference between Q and Q. So imagine that we have a phenotype in grams. Just suppose that it's sugar, for example. So we have the value in the homozygous A2, that is 6 grams, the value of the homozygous A1, that is 14 grams, and as I mentioned, under an additive model, uh, we want to estimate which is the midpoint between those homozygous line. So the midpoint is 10 grams. So that means that we have the same difference between that midpoint and both homozygous, that is 4. So with that, we can estimate what is the value, which is the value of A. And here, as you can see here, the heterozygous lines has a value of 12 grams. So that means that the difference, dominant deviation of this gene, it's 2 grams, because it's the difference between the midpoint and the heterozygous lines. So now with that, uh, just suppose that we have two different populations. So we have the population 1 with allele frequencies of 0.9 and 0.1, and the population 2 with the allele frequencies of 0.6 and 0.4. So if we use this formula to estimate the gene substitution effect, you can see that A is equal to 4 plus the dominant deviation, it's just the difference between the midpoint and the heterozygous lines, it's equal to 2 times the difference between Q and P. And that allele substitution effect in with these allele frequencies is equal to 2.4. And if we have different allele frequencies, you can see that using exactly the same additive effect and dominant deviation, we can get different results. And I just want to point out that the allele frequencies are important, and that is why in different populations you can estimate different allele effects. So yeah, always that effect of the alleles depend on the frequencies that we can find in the population. So just to be sure that everybody understands this concept, because it's really important, I'm gonna try to use another example that maybe is easier to understand. So here in this case, we don't have deviation, a dominant deviation, so the heterozygous lines are just in the middle. So the phenotype of the heterozygous lines are it's 10 grams. So that means that D is equal to 0. And we're going to use the same formulas to estimate the allele substitution effect and to estimate the effect of the alleles A1 and A2. So as you can see here, and also I'm gonna use a different allele frequencies again just to show you the concept. So if we use this formula to estimate the allele substitution effect, you can see here that the difference between the homozygous at the midpoint is the same, it's 4. But now d, that is the deviation, dominant deviation, it's equal to 0 because the heterozygous are just in the middle between both homozygous. So that means that this formula here, the allele substitution effect, it's equal to 4. And if we want to estimate the effect of alpha 1, we just use this formula here, that is q times alpha, and q is 0.5 times alpha. It's alpha is 4, so that it's 2. And here we have a negative value, and remember, as the heterozygous lines are in the middle, so we just identified a that is positive for, in this case, a1, and for the another homozygous is just negative. So that is why we have a negative value here. So the value of alpha 2 it's equal to -2 or -2. And the difference, and the allele substitution effect is equal to the difference between alpha 1 and alpha 2, and that if we subtract a negative value, so that it's positive, negative, and negative is positive, so that is equal to 4. So I just want to show you that both formulas are the same. So what this means? This means that if we increase the number of A1s in the genotype, we're going to increase the population phenotype in 4 grams in this example. So as you can see here, we have for the homozygous A2 6 grams, and if we increase one A1 here, so the phenotype increase in 4 grams, and if the genotype have the 2 A1s, now the phenotype is 14. So that it's the importance to understand the allele substitution effect. Again, so the allele substitution effect it's equal to 1/2 of the difference between the homozygous, so it's the midpoint between both homozygous genotypes, plus the dominant deviation and the difference between the allele frequencies. So if the dominant deviation is equal to 0, so alpha it's equal to the additive effect. And as you can see, we were lucky in the examples because we got all the data and was pretty easy. But if we don't have the data, we can estimate alpha doing linear regression. So linear regressions is a common approach to estimate the alpha values. And as it's common, and the linear regression is useful to estimate the substitution effects, that doesn't mean that the models are perfect. So as you can see here, the black points are the real genetic values, and the white points are the estimated genetic values. So under an additive value model, we can take into account for some dominance, for some degree, in some degree we can account for the dominant deviation. And as you can see here, the models, the linear models do a good job to capture the effect of the genotypes in the phenotype. But that doesn't mean that the models are perfect. And it's, for example, it's a common phrase that there is not models that are perfect, but there are some of them that work. So the linear models work pretty well to capture the additive and in some degree capture the dominance variance in the gene actions. And after identify the allele substitution effect, we can estimate the breeding values, and the breeding values basically is 2 times the alpha 1 or 2 times the alpha 2. So that depends on the genotype, of course, but my point here is that the 2 means that we have two alleles, so that is why we have to use this number here. So because we have in the progeny, if we are talking about breeding value, that is basically how good it's one genotype to produce the offspring. That genotype is just going to transmit one allele to the population, and the population is going to have two alleles. So that is why we have to take into account for the two alleles here. And as a short review, I just want to that you keep in mind three important concepts. So first are the genotypic values. So remember that we have the genotypes, but now we have the additive effects and the dominant deviation. And this dominant deviation is the difference between the heterozygous phenotype and the midpoint between the homozygous. That linear regressions are or have a good or can help us to understand the effect of the genes, and in some degree they can capture the deviation due to the dominance, but they are not perfect. There are some models that are more complex that try to include dominance and epistasis. We're not going to talk about those models in this course, but the most common approach is the linear regressions to predict the gene actions or the effect of the alleles. And, and then for the gene substitution effect, remember that basically we try to take into account the additive effect plus the dominance plus the allele frequency. So the allele frequencies, as you can see here, are really important. So always think that if you have population with really different allele frequencies, it's going to be difficult to use genomic selection. So that is why we have to think about strategies to combine those populations or to try to don't predict one population with another one that is completely different with completely different allele frequencies. So this is the end of this video.